Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Ashley Reaver, and I'm joined by Dr. Gil Blander. Welcome to Longevity by Design, How to Live a Longer, Healthier Life. We're produced by Insight Tracker, your science-based guide to optimizing your body from the inside out. Our guest today is Dr. Nan Hao. Dr. Hao received his PhD in biochemistry and biophysics from the University of North Carolina at Chapel Hill. He was a postdoctoral fellow with Timothy Elson at the University of North Carolina and Aaron O'Shea at Harvard University's Howard Hughes Medical Institute. The goal of Nan's lab is to understand how network architecture governs the dynamics and function of regulatory responses in the context of stress, aging, or disease. His lab has three main focuses: decoding the dynamics of stress responses, probing the causes of cellular aging, and quantifying the heterogeneity in cancer cells. Thank you so much for joining us today.
Thank you.
Welcome to the show now. It's a pleasure having you here with us, Nan. And I would like to start with a question about your background. What made you to become a scientist, and specifically, what made you being interested in molecular biology and specifically aging?
Okay. Yes. So I have been interested in science from very, very early on. Even in my high school and undergraduate, I've been very interested in science and especially in biology. So now looking back, I didn't consider other paths. This is like the natural path to go forward, and it has going well. So that's basically how I get into science. And for molecular biology of aging, I want to talk a little bit about my training. During my PhD from very earlier on, I have two mentors. One is experimental biologist, one is computational biologist. And basically, we through collaboration with them, I obtained this combined trainings in experimental and computational biology, starting from my graduate school. And then during my postdoc, I also did this multidisciplinary work and combining the computational modeling and the molecular experiments together, try to help us to understand how cells operate in the changing environment to achieve their responses. So, and then I started my own lab at UCSD. I started with some studying stress response at the beginning because that's my postdoc work. I continued that work a little bit, and quickly I got interested in cellular aging. And very fortunately, I found some of my colleagues are also very interested in cellular aging, and we form a team and we collaborate together. Has been already for 8 years. And this team is a multidisciplinary team. We have bioengineers, we have physicists, we have biologists, we have computational modelers, but we all share the same interest in cellular aging. So this has been very productive, very interesting, very fun. So that's how I got into the aging field and start working on aging. And so that's pretty much it. It has been fun. I enjoyed doing research in aging.
That's cool, and I'm sure that it was fun. And doing science is fine sometimes. Sometimes it's very hard, but I'm happy. I'm happy that you're saying that it's fun. But to take it to a more serious note, when I bump into your recent paper in the Science magazine, I was blown away about what are the opportunity based on these papers. And instead of looking at one protein or one gene and seeing how it's influenced the lifespan of the organism or cell, you took a much more holistic, network-wise process. So we will dive into that and spend the next, maybe half an hour to an hour in discussing this paper. But let's start building the stage for that. And specifically, I would like you to explain to our audience, if possible, what are synthetic networks and what can you do with them?
Yes, definitely. So probably we can start it with what is gene regulatory network, what is endogenous gene regulatory network. So actually, in our cells, or in all the cells, basically, there are a type of proteins called transcriptional factors, transcriptional regulators. And what they do is they regulate the expression of the other genes. And they can also regulate each other, and this will form a network. This is called the gene regulatory network. So basically consider, let's say, there is transcription factor A, transcription factor B, and the transcription factor A can induce the expression of transcription factor B, and the transcription factor B can repress the expression of transcription factor A. So this is a very simple network. This is called a negative feedback, right? So, and there are other interactions that could be possible, kind of connections to transcription factors that will shape the expression of genes. These are called gene regulatory networks. And basically, almost all our biological functions regulated by certain type of biological gene regulatory networks. So that's endogenous. We have that in nature. And what we can do in synthetic biology really is to revere. First, we can build new networks. We can use the components that never exist in this cell, but we can introduce it into this cell, and we can connect them together into a certain type of network that we can design so that it can do some specific functions that the cell cannot do without this network. That's one way that we can do it. This is also called synthetic gene network. And the other thing we can do it is we can build on endogenous gene network, and we can change how they interact to each other, which is in our current work, this is what we do. We can revary it. We can change their interaction so that this will generate new function that the natural system doesn't have. So that's basically what we can do with synthetic biology.
Excellent. And specifically in this study, you use something that you call oscillatory gene network. So can you explain what does it mean? What is the oscillation and how that how unique is that and so on?
So that's right. In this paper, we build this oscillatory gene network. So the gene network connection for that is called a delayed negative feedback. It's pretty much similar to the example I just raised. This gene A will induce gene B, while the gene B will repress gene A in return. So this is a negative feedback. And with appropriate parameters, this network will be able to generate oscillation. And then this is called the oscillatory gene network. Also, by oscillation, what I mean is the oscillation in the components of this network. For example, this gene A and the gene B both are transcription factors. They will oscillate. Their expression level will periodically change over time. And this is called oscillation. And this network is called oscillatory gene network. So, so that's pretty much what we did in this paper. We revered the endogenous, a little bit of background of the endogenous gene network. We found this, there are two important longevity factor. One is SIRT2. We will get into that in detail, but one is SIRT2, one is HAP. These two factors in the natural system, they inhibit each other. Basically, A will inhibit, SIRT2 will inhibit HAP, while HAP will also inhibit SIRT2. And this is called a mutual inhibition network. This is also a type of network. And what it can generate is a toggle switch. Toggle switch meaning that when SIRT2 is high, then it will repress HAP, HAP will be low. And this cell will stay there because of this mutual inhibition. Because when HAP is low, HAP will not be able to repress SIRT2. SIRT2 will keep going high, HAP will keep going low, so that always be that state. That's a switch. And then if HAP is high, SIRT2 is low. And SIRT2 will never be high again. And HAP will be always high, so that this is another switch. So this is the mutual inhibition will generate this kind of toggle switch. Either SIRT2 is high or HAP is high, but you cannot have both high. Okay, so that's the natural system. And what we found is really the genetically identical cells in their cell either during the aging period, either SIRT2 is high, HAP is low, or HAP is high, SIRT2 is low. So in either way, it's bad because if SIRT2 is high, HAP is low, SIRT2 is per longevity, but HAP is also per longevity, also beneficial. If it is low, then the mitochondria is messed up. So that cell will undergo this mitochondrial decline aging pathway. You can see it very clearly. And however, if HAP is high, SIRT2 is low, although mitochondria is perfectly healthy because HAP is high, but the SIRT2 is very low. DNA become unstable. SIRT2, the function of SIRT2 is protecting DNA, is to stabilize DNA. And DNA is very unstable, and the cell will undergo this DNA instability pathway towards aging. So basically, there are these two different aging pathways in same type of cell. Just depending on which is high, which is low, a single cell will go on one of the single pathways towards cell death. And so that's why we imagine, okay, this mutual inhibition drives this divergence in aging response. How about we make it into a negative feedback instead of the mutual inhibition? That's what we did. We changed the negative inhibition into a positive regulation genetically, so that we revert this gene network from a mutual inhibition network to a negative feedback network. Yeah. By doing that, we start generating oscillation. So it is no longer a switch. It's no longer that one is high, the other is low. It's always there. The cell will die. Because they will keep switching. They will keep periodically cycling between high and low, these two factors. And the good thing about it is that you are not into a state that is detrimental. Either state is bad, right? So these two factors are both important. But if you keep cycling, then the cell will not decide on which aging pathway will go. It will switch between the pathway, and this will dramatically extend the lifespan. So their damage accumulation become slow down, and the cell lifespan is dramatically extended. Sorry for the long explanation. That's basically the idea of generating an oscillation in the pathway.
And you said this was natural, obviously. Are there other natural endogenous oscillatory gene networks that are also exist in humans that people might be used to?
That's right. So first, in our work, this is not natural, right? I mean, this is built it into oscillation. But we build it because we see the nature, how the nature works. We see the oscillatory gene network in the nature, so that we can think of how to build it, right? So come back to your question, are there endogenous oscillatory network in nature? Definitely. In humans, in other cells. The best example is circadian clock, the gene regulation network that underlie our circadian clock, 24-hour period of our clock. Our gene expression, some of the genes are regulated with this 24-hour period, right? And there are a gene network that underlie this very, very rigorous 24-hour period changes. And this is called a circadian clock gene network. And that's a negative feedback network. So, the other classic example is cell cycle. So the cell division is also regulated by this kind of network that will periodically generate another cell. So, and we learn from the nature, basically. We look at them, we see what kind of network can underlie these oscillations, and we do the model simulations to use computer to simulate if we connect the pathway this way, what will happen? And then we can do the experiment.
Awesome. Thank you. And now I'm going to go back to your research. You mentioned SIRT2 and HAP.
Yes.
For individuals that don't necessarily know what those are, could you go into a little bit more detail describing what they are and how they're related to longevity?
Definitely. Both SIRT2 and HAP are like pro-longevity factors, pro-longevity genes. SIRT2 is basically encoded. SIRT2 is Gil's favorite gene, one of them. So SIRT2 is encoded by the arguably best studied longevity gene to date. It functions as a lysine deacetylase. What does it mean is it modified the DNA, the histone on the DNA, so that it can protect the DNA, and also it can regulate the gene expression. And the gene expression regulatory role is more of a repression. It's called protein silencing. It basically will shut down the gene expression for certain genes. And also it can protect, at the same time, it can protect the DNA from instability. This is the major functions of SIRT. And for HAP, it is also a transcriptional factor, transcription complex, actually. It is responsible for the genes that are important for mitochondrial biogenesis and mitochondrial function. So basically, SIRT2 is important for our DNA, the stability of our DNA, important for our genetic information center, let's say, and HAP is important for mitochondrial, our energy production center. So both are very important, and both are pro-longevity. By pro-longevity, meaning that if you delete them, cell will live very, very short, so that they are important for the longevity. However, at the same time, which is important, that if we overexpress either of these factors, it also has some negative effect. So basically, both SIRT2 and HAP, you have to keep optimal range of expression level, which is favor the longevity. So that's actually the basis of our idea. So by using the oscillatory network, we can keep both SIRT2 and HAP oscillating within this optimal level, so that they don't go too high, they don't go too low, so that they can keep functioning to promote the longevity. So, yes.
Yeah, and just a side note, we had for our audience, we had a few discussion about SIRT2, mainly the homolog or ortholog of SIRT2 in human or in mice. We spoke with David Sinclair, Chaim Cohen, Raul, and other scientists that studied aging or SIRT2. Actually, ID Tissenbaum, we discussed about how SIRT2 extend lifespan in C. elegans. So we had many episodes about SIRT2. Anyone that wants to learn more about it can speak about it for a few hours. And we have an exciting subject to discuss, but anyone that wants to refresh his or her memory are more than welcome to go back and listen to those episodes. I want to now try to dive down and try to understand. So yes, SIRT2 extend the lifespan by itself. HAP extend the lifespan by itself as well in yeast. My question is, why have you decided to look at the both of them together? Any reason why you decided to look at the SIRT2 and HAP and not the other longevity in yeast?
So that's a great question. So let me think about how to answer this question. So we are looking at the other genes as well. But why we start with SIRT2 and HAP? So actually we start with SIRT2. As you said, that SIRT2 is like the best studied longevity gene. Everyone is interested in SIRT2. So we started with SIRT2, our cellular aging study, and we designed a reporter that can look at the activity of SIRT2 in the single cell level, and then we track what we have is we can track the aging process of each single cell in yeast, and we can look at how SIRT2 change, how chromatin silencing change, the expression change during the whole aging process. We start with that, and what we notice that here actually I want to also highlight one of our approach, because for yeast aging study, the traditional approach is really the yeast aging is replicative aging of yeast, which is measured as the number of mother cell divisions before its death. And this is a genetically tractable model for the aging of mitotic cell types in mammals, such as stem cells, epithelial cells. And the way to study it, the traditional way to study it is we have to put the mother cell onto the petri dish and look at them under the microdissection microscope. Gil must know that. And then when each mother cell generates a daughter cell, then we count, this is the daughter cell number 1 of this mother cell, and we remove the daughter from the mother cell using a needle. And the reason to do that is if you leave the daughter cell there, the daughter cell will become another mother cell, and they will bear their daughters, and this exponential growth of a colony of cell. You will quickly lose track of which is mother, which is daughter, how many daughters this mother generates. So you have to really look at each single mother cell and then remove the daughter cells every time it generates a daughter cell. So basically, let you sit there for 3 days, every 90 minutes, remove a daughter cell. This is extremely laborious and time-consuming and low throughput. And more importantly, you cannot see the molecular changes because low-resolution microdissection microscope, you basically cannot see any molecular or cellular changes during age. And what you can do is you can generate mutants, and you can measure the lifespan of cells. So what we did is we designed this microfluidic device, which is very tiny little chips, and it has many, many small finger-shaped chambers where these chambers will trap each of the mother cell inside of this chamber. And then the mother cell can still generate the daughter cells. The daughter cells, there are medians flow through so that the daughter cells will be pushed out from these chambers. But the mother cells are stuck here always. This way, under the microscope, time-lapse microscopy, we can look at individual mother cells and take a movie of it for their whole aging process. And we can look at hundreds of mother cells at the same time. So in this way, we really can see if we put in the fluorescent markers or reporters, we can really see the molecular changes during the lifespan of many, many mother cells all at the same time. Using this approach, really, we study the SIRT2 at the first place. We start with SIRT2, we put in a reporter, but what we see really, so it's very interesting, that as I said, genetically identical cells. These cells are exactly identical, the same DNA, the same environment, but they age different. Some of the cells show this SIRT2 become, activity of SIRT2 become decreased during aging process, DNA become unstable. And however, there is another part, another half of the cells where their SIRT2 is perfectly good. SIRT2 activity is very high, the DNA is very nice, but they have some defect. They generate these very tiny daughter cells, and they look unhealthy, and they actually have a shorter lifespan. But they look very different from the other populations, aging cells. So, and we have been thinking, this population, we can clearly see SIRT2 is declining. The DNA is unstable. So this is, the aging is driven by this instability of DNA. However, this half of the cells, their DNA is perfectly good, and the SIRT2 activity is high. What's going on with them? They die from a different, and then we look at different markers, and later on we find out, the mitochondria is messed up in these cells. And what is the major regulator of the mitochondria? HAP is the major regulator of the mitochondria. That really drew us to focus on HAP as well, because we see in the same population, half of the cells die because their SIRT2 is decreased. Other half of the cells die because their HAP is decreased. So these two factors really, they play different role, different single cells. And as I said, then later on, we figure out these factors interact to each other. They inhibit each other, so that this will determine a single cell whether it goes to one this aging path or the other aging path. So this is a very long answer to this question, but this is some of the background of our study.
And this pathway, as you mentioned, it's not something that we only find in yeast, but specifically in cells, meiotic cells like stem cells are one reason to study it in yeast. Can you make it? What's the connection, I guess, for individuals, or let's say humans, taking maybe what you find in yeast to potentially being able to apply that to something like a stem cell?
Definitely. So both of these two pathways are deeply conserved in eukaryotes. So, and as Gil just said, that SIRT2 and the mitochondria, those are very important factors in human aging, mammalian cell aging. Definitely, people already identify that. Actually, when our first paper, the paper about this two aging mode, two aging phase of isogenic cells, are published also like 3 years ago in Science. Danica Chen called me. Gil must know Danica. Yeah,
I know her very well. She used to be my baymate at the lab.
That's right. Yeah. So actually, I presented this work, conference, and later on, Danica reached out to me. Danica is working on this hematopoietic stem cell in mammals, and she said that we should study these two pathways and their interactions in these human cells or in mammalian cells, in stem cells, because the pathway look the same. So, and both of them are very, very important. So there must be some interactions. We should use the same approach to study it. So that's how relevant this is. So, from her perspective, these are the things. So very similar, at least. I think that's our next step. We are talking with Danica and with other researchers to explore this direction.
Yeah, and Danica was our first guest in the podcast. Like
I saw her.
So yeah, we are closing a circuit, in this discussion. Yeah, so now let's talk a bit about what was so can you discuss a bit about what was the effect of this network on the longevity of the yeast cells?
In fact, yes. So that's the exciting part, because it is not surprising that by revarying it can oscillate. I mean, if you do it correctly and you are lucky, they can oscillate. So what is really important is that this oscillator really extend the lifespan. So although we hypothesized that, but it was nice to see it really come true. So really, the oscillation leads to an 82% increase in lifespan in yeast compared to the control cells. And this is the most pronounced lifespan that we have ever observed in yeast, in our hands at least. So actually, we compared the oscillators with the other long-lived, previously identified long-lived single or double mutants previously identified from genetic screens. We compare them in parallel using our experimental approach, our cell genetic background, and our oscillators really can live longer than all of these long-lived mutants, which is very satisfying. So really, this by generating these oscillators, by playing this oscillation trick, we can generate super long-lived yeast cells, way longer than those from the unbiased genetic screen. And come back to your comments you mentioned about previous study. We focused on one single gene deletion overexpression, and why this study is more important, why we need to understand the network. And I think this is, if you understand the network, you can achieve even better results. You have a bigger effect. You can fine-tune it, right? So that's
Yeah, no, absolutely. And I think that's an important take-home message for our audience. So before that, like overexpression of SIRT2 gave on average 30% or something like that, suddenly you have 80%. That's a huge increase. And if we apply this method, hopefully in the future, mammals and humans, theoretically, it can increase significantly the extension of lifespan that we can use by using genetic manipulation or treatment with a drug, or even a mix of a lifestyle intervention. That say that there are some pathways that they work in a different ways, and then if you add them one on top of each other, you have like a layer cake that can actually hopefully will allow us to live longer. That's why I'm so excited about this study. It's really impressive. 82% is really impressive. And another side note that when I read carefully your paper, I've seen, if you look, because not all of that perfectly. So you said that when you separated the perfect oscillator E cells, you go to an extension of up to 105%, which is more than 2x increase in lifespan, which is really, really impressive. I think that the only example that I know is what Cynthia Kenyon done in the 90s on DAF2 and DAF16. That was in this level. But again, it's a different organism that is not replicative organism. And I think that that's the highest increase of lifespan. So that's very impressive.
Thank you. Yeah.
Yeah, Ashley.
All right, I'm going to pivot us to the next topic unless you want to ask any follow-ups in there.
Yeah, I think that we summarize it well, but maybe we'll summarize it again because it's really exciting. When you use a network of genes and you allow them to regulate each other and you engineer it in the right way, you can increase the lifespan much more significantly than when you use one gene pathway or one system. And maybe the application for that in the future, again, it's a model organism. We need to be very careful, but the implication of that can be enormous for our lifespan. And when we interview in the past some of the leader scientists, some of them say, hey, we can live for 300, for 400. Maybe this method will allow us to go there. So this is, that's why I see it as a revolution, because you can add one or on top of each other and another one maybe, and maybe in the next Nature paper or Science paper, we'll have 4 different pathways, and then suddenly it will be 200%, 300%. So we can get to a place that we cannot imagine today based on only one gene or one protein.
Mm-hmm. Thank you. Yeah, that's a very nice summary. That's a good future.
Yeah.
The enthusiasm from both of you is also making me very excited about it.
As a Longevity by Design podcast listener, you understand the value of improving your health for today and for all the years ahead. And if you want to live your healthiest, longest life possible, you need to understand what's going on inside. At Inside Tracker, we take a personalized approach to health span optimization that eliminates guesswork from your wellness plan. Inside Tracker analyzes blood biomarker and DNA data, along with physiomarker data from fitness trackers like Oura Ring, to deliver personalized food, supplement, lifestyle, and exercise recommendations that allow you to take control and improve your health span. And for a limited time, Longevity by Design listeners can get 20% off at the Inside Tracker store. So if you're ready to receive a personal health analysis and data-driven wellness plan to optimize your body for the long haul, then it's time to start inside. Visit insidetracker.com/podcast to get started today. That's insidetracker.com/podcast to get started today.
All right. So your lab investigates the organizational structure of cellular networks and how their organization contributes to processes like stress, aging, and different diseases. In your research, you also investigate causes of cellular aging. So can you elaborate a little bit on how organizational structure contributes to aging and what organizational structure means exactly?
So that's so what I mean there is really usually we call it the gene network architecture or gene network structure. This is really come back to the earlier question. This is how these transcription factors interact to each other. Really, there are many ways they interact to each other, and they all have different effects, different dynamics, different outcome. As I said, classic example: A will induce B, but B will repress A. This is called so-called negative feedback. It will generate either oscillation or adaptation. It has homeostasis. A will, and we talk about the endogenous network, right? SIRT2 and HAP, A will repress B and B will repress A as well. So this is called mutual inhibition. This is another so-called architecture or structure, or right, a network motif. And this is basically, if these two genes interact this way, then it is a toggle switch. One is on, one is off, and then it stay there. One is on, one is off, the other way. And it will stay there. It will never change. So this is called a toggle switch. And it's because of the way they interact to each other, because of this network architecture. And there is other like positive feedback loop. For example, A will promote B and B will promote A as well. This is called a positive feedback. and then it will generate bistability. So basically, so look at single cell, there will be two populations have very different response. So people already study this for a long time, and the major goal of our question, of our research, is that we want to identify these network architectures, both in nature, endogenous, but also artificial, synthetic, and we want to study how this will contribute to biological function. In the context, as you said, in the context of aging, which is this work, the context of stress response, and in the context of disease model. Yeah.
Thank you.
And now we also ask our audience to tell us what are they interested in. And what they said, they are really interested in autophagy, specifically how it's relevant to longevity or aging, and specifically how it is fasting. So discuss a bit your viewpoint about that.
Of course. And actually, it is related to work. I think, especially when we talk about fasting, a very popular topic is intermittent fasting, right? I mean, not constant fasting. Nobody wants to do constant fasting, but intermittent fasting, periodic intervals between, right? I mean, and this is, if you think about it, it's similar to the oscillations, right? I mean, periodic. So I think even the universal underlying principles may be the same. So why they work better than the constant one? It may be the similar principle of why the oscillator is performing better. So we didn't, in our lab, we didn't start to study autophagy yet, but we will look into that. But we are very interested in that, and it is actually definitely related to the mitochondrial HAP pathway and the SIRT2 pathway, I believe. So, but we did look at the fasting and intermittent fasting a little bit using our yeast model. I don't know how relevant it is to human beings, but we generate this oscillatory glucose limitation for the yeast. Look at, okay, will that be able to extend the lifespan? Will that be able to extend the lifespan better than a constant calorie restriction, glucose limitation? And what we see in the preliminary result, this is unpublished. Preliminary result is it indeed have a very dramatic effect on the longevity, periodic oscillatory glucose limitation. And also it has dramatic effects on these two pathways, the SIRT2 pathway and the mitochondrial pathway. We have those reporters and we see dramatic changes there. So even without the context of autophagy, if we only look at the other two major aging hallmarks, we can see clear changes already. So basically now we are trying to understand the mechanism. We build a model of it, but I think this is all. This result really suggests it is universal, right? I mean, it is a general approach to test lifespan. So do it periodically. So, and we have to figure out what's the frequency, what's the amplitude. We have to figure out all these dynamics. That's the actually our next aim. And I think starting from yeast is a good starting point because we can do everything very fast. We can do systematic analysis. So, and with that information, we can then go further to the other objects. So that's what we are trying to do now. So we are trying to explore the dynamic patterns of glucose starvation, glucose limitation, fasting, using yeast as a model of it.
Okay. So let me just try to translate it back to humans. So what you can, you decrease the amount or starve the cells for glucose. So they had a very not very low, but lower amount of glucose, let's say in the bloodstream. And what you have cells live longer, and in a part of it, the changing in the activity of HAP2, and SIRT2. Correct? Okay.
Yes. Okay. Good. And doing the dynamic intervention is important, have an effect.
Okay. So the dynamic, you mean when you have the a process, they live even longer with the restriction of the glucose?
That's another thing to try. We haven't tried that yet.
Okay.
What I mean is that we can do this glucose limitation dynamically. So because previously what people do is culture the cell in this constant glucose limitation condition, like color restriction, right? And then see an effect. That's what.
Okay.
That's not what we want to do, right? What we want to do is, if we want to explore the intermittent fasting or fasting at a later age, we can do it dynamically. We have device that we can do whatever we want dynamically, time dependent, so that we can generate this oscillatory input from the outside and see their effect. And indeed, it has a very dramatic effect, suggesting no matter it is inside or outside, this oscillation can help.
Okay. So you basic the intermittent fasting to the yeast, and you show that it's working, which is cool. Great. So we would like to discuss another focus of your lab, and specifically heterogeneity in cancer cells. So maybe let's start by you explaining what does it mean.
So what we study for cancer is very, very specific. It's from my background is as a quantitative computational biologist and a single cell biologist. What we want to understand is really the difference between genetically identical cancer cells, clonal cancer cells. So because most of the variability between different cancer cells is genetic. They have mutations, they have different mutations, so that it behave different. So we want to ask a question that is different. We want to focus on non-genetic difference. So basically, these are genetically identical cells. And they are from a single clone, but they behave differently. Like the yeast cell in aging, right? They are genetically identical, but they behave very, very different. That's where this genetic networks or this dynamic of gene expression will contribute to generate this difference. That's what we are interested in. So that's why we focus on specific clonal cancer cells, and we look at their difference in gene expression and in behaviors. And maybe this will underlie some of their, for example, anti-chemo resistance, right? And the chemo resistance may be different. This is what we study. And specifically, currently we study this and why they are different. The difference is actually because of the intrinsic stochasticity in gene expression. Even the genetics is totally the same. The DNA is all the same. The way they express the gene, there are noise in it, randomness in it, stochasticity in it. So that will be amplified and then end up different phenotype. So that's for sure in many cases. So that's what we are interested in. And currently what we are interested in is this mutation. This is TERT promoter mutation. TERT is telomerase gene, and which is super important. The expression is super important for cancer. If you have too much of it, it will lead to cancer, almost a causal relationship. So, and people identify this foreign mutation in the promoter, a single mutation in the promoter that is highly correlated with the cancer occurrence. So, and what's going on? A single mutation in the promoter. And they look at the gene expression. If, let's say this single mutation is super important for gene expression and you have a huge amount of TERT, then it makes sense. But actually what they saw is they didn't see a huge increase in TERT expression. They see like a twofold increase to the most. And this should not be able to lead to cancer. I mean, it's not that high. So that's the question. And we start our study there. And our hypothesis is because they look at the population level. So they look at the average level of the TERT expression. However, if TERT expression in this mutation, with this mutation, it will be spread out like among different cells. meaning the average will not be increased too much, but the variability is huge. And then some cells will express this high level of gene, third gene that will lead to tumor. And the tumor, you don't need every single cell to be a tumor, right? I mean, only one cell, they will start growing into a tumor. So that's our hypothesis. And that's what we are working on now to look at the difference in genetically identical cancer cells.
So again, to translate it to our audience, what you are saying that if you look at the cancer cells and specifically on the expression of TERT, which is an enzyme that allow the telomeres to be replicated and then allow them to escape the senescent and grow like crazy. What you're saying, if you look at the population of the cancer cells, the expression of TERT is only 2x. But then if you look at the specific cells, you have some that are 2x, some that are 1.5x, but a few that are, let's say, 10x. And because they are cancer cells, they can replicate fast. And those are basically the beginning of the cancer that they can replicate like crazy and basically kill the organism. Is that fair?
Totally. Yeah. This is very preliminary hypothesis. Whether this will really lead to tumor, we don't know. But that's the hypothesis. That's the direction that
processes
we are.
Is the is your thought there that there are specific cells that, just like these cancer ones that overexpress that maybe it's just a couple of cells that make everything age faster or bring up that average, like you said before? What's the overlap that you see between cancer biology and aging processes?
That's right. That's very possible. So as I said, the underlying principle are the same. We both understand the network architecture, how they contribute to the cellular responses, and also at the single cell level, right? I mean, different single cell will behave very differently, although they are identical, actually, genetically identical. So in aging, definitely, as I just mentioned, we see that identical cells can age toward two different pathways. If I look at the population of white type cells, their lifespan can be like tenfold different. And they should be identical. They die at the same environment, same DNA. What's going on there, right? So, totally. I think the basic principles are the same. The noise, stochasticity in gene expression play a big role. And this difference is amplified by gene networks and to the phenotypes.
Okay, we're trying to squeeze so many different things into this because there were so many topics that we wanted to talk about. Next, I want to transition into stress because that's also something that you focus on in your lab. As we age, we know that our body's ability to respond to stressors can change, and that stress can definitely be impactful for aging, specifically that kind of prolonged stress that can really accelerate the aging process. We do know there's different types of stress. Some is acute stress. That's really good. Maybe like exercising is a form of stress in our body, but there's some beneficial aspects for overall health, in contrast to something like smoking or a job loss or any other type of prolonged stress that we know can result in really negative impacts. So how does the body differentiate between various types of stress, and why do some stressors elicit positive responses while other ones have really negative?
Okay. So these are two questions. One is how human body or animal or cell cope with different types of stresses, and then why different stresses may have different effects. So the first question is that at least, again, I have to start from my standpoint, which is the cellular response. And so cellular response really, cells have different pathways to respond, specific pathways. They evolve specific pathways to respond to different types of stresses that we know. For example, the oxidative stress, there is oxidative stress pathways that are coping with it. And there is osmotic stress pathways, heat stress pathways. And these are all very different. So cells evolve all these different pathways to specifically confer resistance to different stresses. So, and then come to the other question, why their effects are different. To me, from my perspective, I think the the timing and the stress is different. Of course, the nature of the stress also contribute, right? As you said, that's a job loss. That's terrible. So, well, the other hungry for one meal, that's totally okay, right? I mean, so the nature of the stress definitely contributes. But even let's talk about the same nature of the stress, the same type of stress. But maybe the duration of the stress, the timing of the stress, and the strength of the stress all contribute to their effects, and which may be opposite. So, for example, like earlier life, mild stress. Basically, the idea is it can prime the system to prepare for the future more severe stress, so that it actually have beneficial effect to prepare you, right? However, if it is a very long-term severe stress, of course it will be detrimental. So I think the timing of the stress, the duration, all this will matter for the outcome. That's not only for health, but also for aging as well.
Okay. Speaking about chronic stress causing a specific age-related diseases, or we just accelerate all the processes related to aging?
So again, from my perspective, we can always look at individual animal, individual cells. I want to see the difference between them. What I believe is that different types of chronic stresses will lead to different type of age-related outcome consequences. And aging is not like a common pathway. I believe that in different cells, aging is different. Aging is driven by different molecular pathways in different cells. And that's why if this, for example, if you have protein stress, then this will activate all this protein stress response, and will cause damage in protein homeostasis, and this will lead to aging, and this will lead to this protein stress-driven aging process. While the other stress, DNA damage stress, will drive the DNA instability, and the cell will die in the other way, trigger another aging process. So I definitely believe it is not just accelerating a universal aging process. I think aging is specific at the level of cells, single cells.
How about genetics? Do they contribute to how we process or respond to certain stressors?
Oh, yeah, of course. Yeah. So as I said, that most of the stresses are coped with by stress responsive pathways, specific stress responsive pathways, which their components are encoded by genes, right? So if you have mutations in these genes, this will definitely change the way you respond to stress. So genetics definitely contribute and play a bigger role, pretty big role there. Depending on the context. And from the synthetic biology perspective, we can even revere the stress response pathways, so that we can let the cell to respond the way we want it to respond, not the natural way, but the other way. So that's another idea that we have been thinking, we have been working on.
On that line, can we train, so maybe implement some of those synthetic techniques, the body, and how to optimally respond to stress at a cellular level? Is that, exercise might be a good example, like you mentioned before. A little stress is good, so it prepares you for other bigger stress. But, are there other things that we can do to help the body respond to stress?
Exactly. I think the easiest way, the most natural way, of course, is that, as you said, exercise, or the other mild stress that is not detrimental, is very transient, and it primes the whole system to further stresses, to more resistant to further stresses. So that will be definitely beneficial. That is a kind of training, right? Priming the system. So, and that's doable. So, genetically, we said that we are thinking about synthetic biology way to optimize the cellular responses for humans. But that's a long way to go. So, yeah.
Yeah. And I think that's a very interesting point of discussion. And actually, at Insta Tracker, we are aiming to help people to live a healthier, longer lifestyle. So you already mentioned exercise as maybe a lifestyle intervention that might help you to decrease stress, and I assume also allow us to live better and longer. My question to you, are there other lifestyle intervention that people can use in order to live better longer?
So first, I'm not an expert of that, but as far as I know, in addition to exercise, be careful with your diet, right? I mean, low calorie diet and intermittent fasting. I'm not testing that yet. I'm not trying by myself yet, but I heard it is very beneficial. And probably at some point I will test that. From biology perspective, it seems beneficial, right? I mean, so I think this healthy lifestyle that we know, right? I mean, no smoking and no drug and sleep well and eat well. I think this will all contribute and exercise well. So yeah. So unfortunately I don't have some tricks for that, but I think we just stay healthy, right?
Yeah. I think that nobody has a trick and there is no silver bullet. And what you mentioned, it is what it is. You want to live better, longer, you know, not yet a silver bullet drug that can help you to live to 100. If you want to live to 100, work hard and be on your best, and hopefully you live not forever, but for 100.
And we will try very hard on our side to make synthetic cells to help you.
Exactly.
Awesome. Gil, I think that we answered my top tip for health span. Do the work. There's no silver bullet. Nothing easy until we develop some cool cells that can help you out.
That's right.
Awesome. Well, I think we are wrapping up. We finally got through all those questions. Thank you so much. I learned a ton.
Thank you very
I
much.
would love to have you as a professor. You were so excited explaining so many of these complex topics. Thank you so much for your time.
Okay, so I think we are coming up on our time. Thank you so much, Dr. Hao. We look forward to exploring the research in the field of longevity each month with you and the other leading scientists. For more information, please go to www.infotracker.com/podcast. Again, thank you, Dr. Hao, and it was a real pleasure for being I hope that our listener to learn about the exciting discoveries that you had. And hopefully, very soon you'll have more. I would love to invite you again and discuss those.
I'd love to.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.